Good morning, everybody. I'm very happy to make a presentation here. I'm representing the Albert Einstein College of Medicine in the Bronx, New York, which is not too different from here because a lot of people speak Spanish there. Okay, so I'm going to talk about innate immunity, how innate immunity is an issue for viruses, or it is not, how viruses deal with innate immune functions. So what is innate immunity? Innate immunity is one aspect of the immune system which is ready to act, jump at the parasite or the pathogen, right? As opposed to another system which is adaptive immunity, which takes many days to really kick in. So we want to understand how HIV escapes innate immune functions. So HIV is still around. For some people, this needs to be reminded, and it is a big issue in South America, North America, India, especially in Africa. The numbers are here, but the big numbers are about 38 million people currently living with HIV throughout the world. And my lab has been interested in understanding variation in HIV, genetic variation, and also human genes that aid and abet HIV infection and replication, so that we may be able to come up with a way to deal with control systems that keep HIV at constant replication down so that we can block it. So that's our idea. So I want to introduce HIV properly to you. So HIV comes in 4 flavors, M, N, O, and P, as shown in this slide. And each of these is a single transmission from monkeys to humans. And the M is the most successful because 97% of that 38 million I told you is because of group M. That group M is subdivided into many subtypes, A through J. And each of these form a slice of the epidemic. For example, you can see the big blue half of the circle is subtype C, which is responsible for half of the HIV burden in the world. And that is present only in Southern Africa, Eastern Africa, India, and Southern Brazil. And subtype B is North and South America, but only 10%. And why am I telling you about these subtypes? Is because there are differences between subtype B and subtype C in how they cause disease or how they replicate. So, for example, in my own laboratory, we have shown that subtype C is different from subtype B in the ability of the infected cells to make it to the brain. I don't know how many of you knew HIV infection not only causes immunodeficiency disease, which everybody knows, but also causes neurocognitive disorder. So we showed many years ago that subtype C HIV-1 infected macrophages cannot make it to the brain as efficiently or as fast as subtype B. They hardly go there, actually. So we said that that is a reason why severe dementia associated with HIV doesn't appear to be as common in India or in other places with subtype C. So this has been our thesis. And what this slide is trying to show you is that different parts of the world have different subtypes. So that's another question. Why only subtype B in South America, North America? Why only subtype AA, for example, in some parts of the world? So this question is also a big question for my laboratory that we study this. I want to also tell you that we need to understand our enemy, right, in order to win over the enemy. So this is our enemy. So I want to show you the enemy's lifestyle. So the lifestyle of HIV is that it's a virus with 2 copies of RNA, and that RNA undergoes reverse. It enters the cell, at the cell surface. You see the green line is a membrane. So the virus enters the cell. So that core undergoes conversion of the RNA into DNA, and the core then enters the gate of the nucleus, which is called the nuclear pore, where it can now insert its DNA into the genome and becomes permanently part of our genome, which is why HIV is so hard, almost impossible to get rid of. So that completes the first half of the infection, and then the late events start. mRNA starts being made. mRNA goes to the cytoplasm, where it makes the proteins, viral proteins. They assemble near the membrane, and then they bud out of the cell to release the progeny virions. So one infected cell can produce hundreds or thousands of virus particles. So I mentioned to you that we are interested in host proteins, right? So when this paper came out, we very intently studied this paper. This is a paper from our German colleagues who were interested in identifying an inflammatory gene in the host that may be correlative with HIV infection. So why inflammatory genes? So inflammatory genes are part of the innate immune system. So innate immune system, what it does, it causes inflammation, inviting other white blood cells to come to the site, or they send out chemokines. Chemokines are like breadcrumbs you throw to attract certain types of cells. So different chemokines bring different types of cells. So we found this paper showed that out of the 110 genes they examined, just one gene, its product, correlated with HIV. That was called CCL2, or it's a chemokine of a particular type that attracts macrophages and monocytes to the site of infection. And this was surprising to us because we expected. So the result is that people who have HIV in the blood have high levels of this chemokine. People who do not have a low levels of this chemokine in plasma. So we expected the other way around. We expected that the low virus would be high chemokine, because chemokine is bringing white blood cells to eliminate the virus. So we want to know what is going on with this. So therefore, we did a study. We asked the question, is it possible that CCL2 is actually enhancing virus replication? So that was our first experiment. But we also were interested in, are there differences between subtype B and subtype C? Remember, I told you subtype C is responsible for 50% of the HIV burden in the world. So what is so special about subtype C? So when we analyzed, look at the left side only. So this is subtype B infection of macrophages. And you see about 15 days into the infection, you have the peak. Now you add more CCL2 to the medium, you see the peak goes higher up. Now you add antibodies to CCL2 to block the CCL2 function, so you see the virus all the way goes down, almost hitting the floor, right? So that says that CCL2 signaling through the cell surface receptors is doing something that is helping HIV. What exactly is it is helping with? We figured out that, first let me tell you, the subtype C doesn't seem to care. It doesn't increase, it doesn't decrease when you change the CCL2 levels. So we hypothesized that this means that HIV is not coming out of the cell. So we looked into this, and I'm trying to show you how HIV buds from the cell. So at the bottom, you see a long bar. That is a GAG protein, which is a main HIV protein, which is made upon infection. That protein, through the N-terminus, which is at the left end, attaches itself to the plasma membrane. You can see now small bars at the plasma membrane. So these assemble at the plasma. This is the membrane. They assemble. They start clustering together, form a little mat. This mat grows. As the mat grows, you have a sphere, and finally there is a bud neck. The bud neck has to be cleaved or severed, and that severing is done by a set of proteins called escort proteins. Those escort proteins form a spring-like structure. That spring tightens and causing the virus to bud out of the cell. Okay, so how does that cellular protein get to the place where the virus is assembling, right? So it just doesn't randomly come there. The virus invites it. So there is a way that the GAG polyprotein at the C terminus at the right end, you see the P6, that has motifs. There are two motifs. One is called PTAP, shown in blue, and the other one shown is red, LY. These two bind to different proteins that are part of the escort. So the PTAP binds to escort 1, and then brings in escort 3, which is blue, whereas LYPX binds Alex, which directly brings the escort 3, which is the blue. There are two pathways for the same thing. So what we have shown is that subtype B has both PTAP and LY. And we showed that the LY is required for Alex binding. So this is how it brings Alex to the site of budding, whereas subtype C does not have LY. So we can actually take subtype B, we can eliminate LY, we can make it behave like subtype C because it can no longer bind Alex, or we can take subtype C, insert LY, and make it behave more like B. So this was very interesting because when you add these two amino acids and make a virus, this virus, when you infect, you add CCL2, suddenly makes a lot. You add anti-CCL2, completely blocked, right? So the question was, how does CCL2 control Alex protein? So we have complete mechanism worked out, but I can only tell you the summary because of the time. So the mechanism we have found out is that when CCL2 binds to the receptor CCR2 at the cell surface, there is a signaling mechanism that allows Alex to be released from the actin cytoskeleton network. It's part of the large, there's a cell is held up by a lot of fibrous, filamentous and fibrous structures made up of actin, tubulin, and all these molecules. So actin cytoskeleton is bound to Alex, and when CCL2 signals, Alex comes off. If you block the signaling, the Alex protein dramatically, almost completely reassociates with F actin. So we've shown this. So now I want to tell you, one of the questions I mentioned is that we wonder about is why subtype C is so dominant in the world. So we thought that subtype C must be very, very fit, highly lethal. That's why it infects more people and causes more HIV/AIDS. But it turns out that is not the simple story. So it turns out that if you compare subtype C in green with subtype B in red, you see that subtype C actually replicates poorly. You add 2 amino acids to subtype C, and it suddenly improves its replication in 2 different cell types. At this point, we found that in some parts of the world, like India and South Africa, both of them have subtype C, they acquire a tetrapeptide sequence, which also binds Alex. And that variant, so we created in the lab, and then we studied that. And what we found is that you see that on the top, you see the second, the 2 bands. For BP6, you see 2 bands. The second band at the bottom is the one that shows Alex can bind. So B can bind, C cannot bind. If you insert the tetrapeptide I mentioned to you to either C or C with PYKE or C with PYRE, you see that Alex binding is restored. And you can also see replication. The green line is subtype C. You add back 2 amino acids, it improves the replication. If you add the 4 amino acids, it's even higher and higher, showing that the viral fitness is very low in subtype C, and it's by being very low fit replication virus, it stays quiet in the cell, in the body, so the immune system allows it to replicate longer. More people get infected by HIV subtype C. This is the hypothesis. So finally, I would like to tell you that we have uncovered a key mechanism, namely that CCL2 can move Alex in and out of the active network. So this is very exciting for us because it is known that monocyte migration, I told you that monocyte migration to the brain is deficient in subtype C. It doesn't cause neuroAIDS or dementia. So we want to be able to achieve that. So we have come up with a way of mutating the Alex protein, which, these are the mutations we made, and several of the mutations worked in our hands. And so now we have a mutation. I won't go into that. So this is our final current goal. So CCL2 is known to remodel the cellular actin cytoskeleton, because as soon as you add CCL2 to cells, cells undergo this kind of a change. You can see uninfected cells are round, infected cells become long, so they can invade the blood-brain barrier and go into the brain. So it makes it easier for them when they're elongated. So we think we have a handle. We have generated mutant of Alex that can kill this ability of cells to respond to CCL2. And this, I'm not saying that this is a way we have solved the problem, but we have understood the mechanism, so which tells us that it is possible to prevent monocyte migration to the brain eventually, so that HIV-associated dementia can be potentially reduced. So with that, I want to thank the folks who did the work. David Ajasin and Vasudeva Rao are the two individuals who started this project in my lab, and Dina Moffett is the one who has been really running the show these days. And those are some of the other people. On the right, Ganjam Kalpana is both my collaborator and my spouse, and Anne Bresnik in biochemistry, and Dr. Andras Fischer, also in biophysics. Thank you very much.